In the world of cancer therapy, one of the most promising weapons we've built is the adaptive T cell, immune cells collected from a patient, trained and expanded outside the body, and sent back in to hunt tumors. But while these therapies have changed the game for blood cancers, they often falter against solid tumors. Inside those tumors, oxygen runs low, nutrients vanish, and immune cells starve before they can finish the job. What if, instead of protecting T cells from that stress, we could teach them to thrive in it? In a bold new study, scientists did exactly that, by harnessing one of biology's oldest survival mechanisms, the cellular response to starvation. At the heart of this discovery lies a molecular sensor called GCN2, a protein that activates when cells detect a shortage of amino acids. It's like an internal alarm system that tells the cell resources are low, adapt or die. The researchers wondered, if they could flip that switch deliberately, could they prepare T cells to face the nutrient-poor chaos of a tumor? To test it, they took freshly isolated T cells and treated them with a compound called halofuginone, a small molecule known to activate the GCN2 pathway. This stress training took place ex vivo, outside the body, before the cells were ever infused into a host. The results were remarkable. Instead of weakening the T cells, the induced stress reprogrammed them. Their mitochondria, the powerhouses of the cell, became stronger and more efficient. The cells shifted from burning sugar in quick bursts to relying on deeper, more sustainable forms of energy. They produced higher levels of the molecules that make them lethal to cancer cells, like granzyme B and interferon gamma, and displayed stronger activation signals such as 4-1BB. Deep inside, the mechanism told a story of adaptation. The GCN2 trigger set off autophagy, the cell's recycling system, which then reshaped the CD98 mTOR signaling network, transforming the T cell's metabolism for endurance rather than speed. When these reprogrammed T cells were infused into tumor-bearing mice, the difference was striking. The treated cells persisted longer, infiltrated deeper into tumors, and controlled cancer growth far more effectively than untreated counterparts, in some cases, eradicating tumors entirely. The same approach worked with human T cells in the lab, which adopted the same resilient metabolic profile, hinting that this strategy could translate into real-world therapies. Even more encouragingly, the process appeared safe. No toxic effects, no uncontrolled activation. Just a smarter, tougher army of immune cells ready for battle. The implications reach far beyond this single experiment. It suggests that we can strengthen immunotherapy not only through genetic engineering or new targets, but by fine-tuning the metabolism of the cells themselves. Instead of flooding them with fuel and signals, we give them a taste of scarcity, and they emerge stronger. It's a subtle shift in philosophy, training cells through controlled hardship, the way athletes condition at high altitude so they perform at their peak when the real test comes. There's still work ahead, optimizing doses, ensuring long-term safety, and proving the benefits in human trials. But the principle is powerful. By activating the ancient starvation sensor GCN2, we can awaken a deeper resilience within T cells, transforming them into long-lasting, energy-efficient warriors capable of surviving the harshest tumor microenvironments. In the battle against cancer, this could mark the beginning of a new era, one where we don't just engineer our immune cells, we evolve them.